Okay, good afternoon again. All right, so now that I have the YouTube going, let me go ahead and get the share page up. And I did this with a student yesterday. If anytime you want to, this is my last class of the day, so if anytime you need to stay behind for extra explanation or some one-on-one time, I'm available. This is so after your class on um, Mondays and Thursdays, I'm available to stay extra for extra tutoring or whatever you may need. And I need to post that on the page, but that's my availability time to help whoever needs help. Oops, what in the world just happened? Interesting. Now my whole page just went, my viewing page just went sideways. Okay, so I just want to go over this one again for those of you who might not. Um, some of y'all are doing it, but you might not fully understand why you're doing it. So let's talk about using. So again, backing up a little bit, this is kind of review. The whole issue with DNA is to replicate. The DNA has all the information for making all the structures in the cell and in the body. And so it just has instructions. It never leaves the nucleus. So we have to find a way for the information to get out of the nucleus to the parts of the cell that make the protein. So its job is to make proteins. So we have these two activities: transcription and translation. So what we do when we read the DNA, that's called transcribing, and the messenger RNA does that. So it makes a complete copy, but it's a reverse copy. It's like a negative copy of. So when you're taking pictures and you have a negative and then you have to develop it into the original picture again, that's kind of what the messenger RNA does. It makes a copy of the information that is kind of opposite to what it says. And then it goes to the ribosome where the ribosome is like the little factory, and it lays out and becomes an assembly line. And then the transfer RNA transcribes it, but it actually transcribes it back into the original message. Okay. But in the video that we saw yesterday, we saw that we're not we're not basing our amino acid retrieval on what the transfer RNA has on it. We're basing it on what the messenger RNA says. And we read the messenger RNA in 3 sets, 3 nucleotides at a time. Okay, so we get a codon. And that codon is specific for one amino acid. Okay, now there are several codons that, as we looked at our chart, there are a couple of codons that translate or code for the same amino acid. So like we're looking in the first column, phenylalanine has two ways. Leucine has like 6 different ways to code for that amino acid. Then we have the next one that has 3, and valine has 4. There's only one that starts, so you need to remember this. Methionine is the start codon. It doesn't necessarily retrieve an amino acid. It just says this is where we start, because the messenger RNA is a very long chain. Not all of it is being read at the same time. It reads it in sections. So it starts at a particular section where methionine, where it's coded for methionine, and it says start making the protein from here. Then it'll keep adding amino acids, and they'll link together, and the transfer RNA will release that amino acid and go back and get another one of the same kind, because each transfer RNA is also only coded for one amino acid. It has a triplet code on the end of it also. So it's only going to retrieve one kind of amino acid at a time. All right, so it drops it off. Once they're connected, it goes and gets another one. And it will keep doing this, and it'll keep going down the assembly line. Like we keep looking at ours as being kind of short, maybe 4 or 5 of them, but they're really very long. So it'll read a very long chain, and then it'll get to these in our third and fourth columns that say stop. So you have your triplet codon, and there's 3 ways to tell the chain to stop getting amino acids. And then the chain will break away from the ribosome, and it'll fold, and then it will be sent to where it is packaged to where it needs to go, because the Golgi body is the packaging center. It knows exactly where that protein needs to go, and it will send it there. All right, so the other review is how to read both of these charts. So you have your triplet codon. When you're using the box, you use the first letter for the on the left side. The second letter is at the top. Third letter is on the right side of the box. And the same thing happens with the wheel. The wheel, you start in the middle and you work your way out. So instead of there being 3 sides, there are 3 circles. And you move from the inner circle to the outer circle. And then your outer spokes always code for the will always tell you which amino acids you're going to retrieve. Okay? All right. The other thing that people were getting kind of wrong, and I'm sorry that the worksheet did this. This is actually one long chain of DNA. So this ACT should have been on the end, and the ACT should have been coded for G A G G A no, sorry, U G A UGA. How can I forget that one? And the UGA, if you go back up to your chart, is going to be the stop codon. All right, so on that first row, you should have done all of you should have a straight line of RNA, and then you need to go back up to the chart and figure out what amino acid is being asked for by that particular triplet codon. Notice it only shows you the RNA. It's not showing you the transfer RNA. The other thing you need to know about the triplet code on the tRNA is that's called an anticodon. All right, so that takes care of that old assignment. That was kind of a review. Now let's look at the mutation one, the one that most of you owe today. It's easier for me to go back to the home page and pick it up. And for those of you who can't find it, the assignments are always on your to-do list. Oh, actually, I don't see it does that to me sometimes. All right, this is how I can get around that. Sometimes when I click on it from the page, it takes me to somebody's assignment. All right, so for today, we want to do the same thing. We're looking at the fact that the protein that we're making this time is hemoglobin. So again, when we make these long chains of amino acids, they fold. So it looks like this little bunched up thing up at the top. And this particular one that was made is the model of the protein hemoglobin, which is found in your red blood cells. Okay. And The DNA strand is our first row up at the top. What they wanted you to do is create an RNA strand. And then once you've created the RNA strand, you're going to use the chart. And if you need to go back to the chart on your other worksheet, do that. Otherwise, use the wheel to figure out what amino acid should be in the bottom row. All right, then you want to look carefully at the fact you're going to do it again. This time we know that we are creating, there's a defect on the hemoglobin molecule or protein, and that's what causes sickle cell. So when you do this second row, notice very carefully that one of these genes triplet codes is not the same as the one above. And when it's not the same, it's going to create a different code on your messenger RNA, and then the messenger RNA is going to tell the tRNA to get a different protein. Sorry, a different amino acid. Okay? Now, the effect of that is that a regular one makes a regular shaped red blood cell, and the defective one or the mutated one makes a sickle shaped red blood cell. Okay. And I think yesterday I told you when you answer this question, what is the relationship? You need to explain how the DNA makes a codon. So it transcribes first to the RNA, and then the RNA is read in sets of 3, and that's the codon. So the codon starts in the RNA, not the DNA. So the DNA is read or transcribed by the messenger RNA. Then the messenger RNA goes to the ribosome where it is read 3 bases at a time, which is your codon. And those 3 bases are what make up the protein or make up an amino acid, which makes up the protein being coded for. Okay, so you kind of have to say all that. All right, so that you fully understand it's a process. Because that question says, what is the relationship? So the relationship is the DNA talks to the RNA. The RNA creates a triplet code which talks to the transfer RNA to tell it what amino acids to get. And then the amino acids form a protein chain. Okay? So somehow, and I just said that 2 different ways. So you pick which way works best for you, but that's how you need to explain the relationship. All right. Next one wants to know, how does the shape of this protein relate to its function? It says be specific to this exercise on sickle cell anemia. So again, we need to know what is the function of a red blood cell, and that the shape that when we need to explain that the round shape allows it to do that function. The sickle shape does not reduces its ability to do that. And I'll tell you again that the function of a red blood cell is to carry oxygen. When it's shaped properly, it can carry a lot of oxygen. When it is misshaped, it carries less oxygen. So if you notice, if you know anyone who has sickle cell anemia, they have a tendency to need to be on oxygen sometimes because they get out of breath and are not getting enough oxygen based because of the fact that their cells just don't carry enough oxygen. All right, again, this paragraph down here, these are therefore a clue that's not multiple choice. You have, what are these 4 things called? Is the name that goes in the space, then complete the rest of this paragraph and you're done. Okay, so if you're done with that, you get that submitted and you don't owe any other work. Please go back to the module. Read the exercise on mutations, because that's what this exercise was about, mutations. I've opened up the technology one, so just please read these. And Monday, when you come back, we'll talk about technology. We'll incorporate the Halloween exercise that you did, because that's part of technology. But when you do types of mutations, make sure that you do this challenge, things that you should do before you go. Do the challenge, do the challenge. All of these things will be on the quiz you'll have on Wednesday. We'll come back. And then after Wednesday, we're not doing anything new. We're going to review for the final exam, which is a semester exam. So it'll cover everything we've done from the beginning. All right, so once you've done the challenge before you go, make sure you read the questions also know before you go. Okay? And we'll deal with technology when we come back. Any questions? Any questions? Please ask questions. If you don't understand, if you still don't understand, make a note to yourself who matches who. Always know that on a DNA strand, A will always match T, G will always match C. And you need to know that for the complementary. When you're making an RNA strand, so the DNA may have a T, and on the RNA, it still will be an A. But if the DNA has an A, there are no T's in RNA. So on the RNA strand, you will have U. So for RNA, it's always going to be A will match U and G still matches C. Okay, so if you code that wrong, that's where you get the mutations. If you try to code it like it's a DNA and try to put T's in there or use where it doesn't belong, it reads what you put. So if you do that, it's going to give you an amino acid that's not supposed to be there, and this is how we get mutations. Okay, so again, if you need to ask a question, please do so. All right, so there's no new work other than that. Please make sure that everything, I'd rather you do a late assignment than leave the zeros in the grade book. We are syncing the grades on Friday. I had to go in and take out the zeros on the extra credit one because that was causing Infinite Campus not to sync with Canvas. So we're working on that. Because some of you have a better grade in Canvas than you do in Infinite Campus. And so we're trying to fix that and make sure that they are the same. All right. So that's my job. You do your job and we'll all be good. Again, while I'm doing that, ask questions. I am watching the chat. If I happen to not catch the chat because I'm so busy grading, just come off mic and say, Miss Harris, please check the chat. Again, a late grade is better than a zero. Please try to catch up on all assignments that are missing. I do have to override. Canvas will take off so many points for late assignments. So in order for some of you to get a grade and it not be zero, even if I grade it, I have to take that late off, but I still will write late in the text box to let you know you didn't get full credit because it was late. Okay, but again, a late assignment is better than no assignment. Because at the end of the day, you still have to know, for the final exam, you still have to know all these topics. The worksheet, go to your to-do list on your homepage. Um, No, if you resubmit, if I said resubmit, no, it's not considered late. In fact, I think I have to go in and take, I think I had the date for yesterday, so I have to go in and take that off as the end date, and I'm going to change that date for tomorrow. So let me get to that too. But no, a resubmit is not late. Mm, okay. Andrea, did you find the worksheet? Okay. Go to your home page. On your to-do list, it should be the one that says mutation. Hold on, let me see what exactly does it say. It should be the last thing on your to-do list. So click on that. You don't have that one? Okay, hold on 1 second. Let me screenshot this again. Um, my computer is so slow. I'm still trying to pull it up. My computer from changing from one page to another. Or I should say DNA proteins, HBA mutations. That's what it should say. All right, I don't know why this keeps changing directions on me. All right, so it should say that DNA to protein HBA mutations. That should be, and it says view it as a student. So let me view as a student. Okay. Failure to load to-do list. Why is that? That's what the issue is. I wonder, let me see if I can't refresh that. Okay, there it is. Refresh your page, because that's what happened with mine. Had to refresh the page. Oh, I refreshed it and it went back to, okay, I'm as a student. Okay, there it is. All right, so this is the student view. So if you click on the last thing on your list, it should be the DNA to protein mutations. So refresh, you might need to refresh your page because it's going wacky wacky. Let's see if that helps. Because I know mine just said failure, so maybe it's doing the same for you. Well, let me know if you get it. It's still not there. All right, how about if I just send you this link, copy link, and let's see if this will work. All right, try that. I sent you a link in the chat. Okay. All right. Let me know if you need the other one as well. Yeah, some wacky things have been going on. I had another student who couldn't upload their assignment. I had to find another way to get it to upload. Canvas just wasn't receiving it. So let me know if you run into problems. And if I can be of help, I will. That one I had to ask Mr. Phillips to help me with. I'm sorry, I was on mute. Okay, let me back up. I sure was doing a great explanation. All right, what I said while I was on mute is that it is important. We're talking about question number one on this worksheet. And I said that um, being as you all have, this is the next class that takes an external exam at the end of the year. So it's important that you answer a question the way they ask the question. So if they ask you for what is the relationship between these three words, they don't want you to define DNA, define codon, and define protein. Okay, they want you to say how one word is linked to the next word. So what you want to say for this question is that the DNA is read by the messenger RNA. And then it takes that message to the ribosome where the message is read 3 bases at a time. Those 3 bases are called a codon. So now you've linked your DNA to codon. Now we have to say the codon codes for one amino acid. And when several amino acids are linked together, they make a protein. So now you've connected codon to the word protein. Okay. So that's how you have expressed there's a relationship starting with the DNA. The DNA's end design is to make a protein. It does that by telling the RNA what to do. The RNA reads it. Now we want that word transcribe. It goes to the ribosome where it is translated 3 units, 3 bases at a time. So those 3 bases are called a codon. Each codon then codes for one amino acid. The amino acids are linked together and they make a protein. Okay? All right. So like the first worksheet, I didn't even have to look at if you coded the RNA correctly. I looked at what amino acids you retrieved. If you didn't retrieve the right amino acids, then I had to go back and look at, did you code the RNA correctly? Because if you don't code the RNA correctly, it retrieves the wrong amino acid and then it makes a mutation. So that's what your activity today is. How did we get a mutation? Because a wrong amino acid was, it's not that it read it wrong, it read what it said. The DNA somehow had a um, a misprint, and we'll talk about um, translations later. Well, we kind of talked about it when we did mitosis and meiosis. We tried crossover, and sometimes it changes the arrangement of the chromosomes on the strand. And if it changes the arrangement, then the RNA will read what it says, and then it will retrieve the RNA, the amino acid that it's said to retrieve, even if it's the wrong one. If it retrieves the wrong one, it makes a mutation. Mm, all right. That'll work. Amazing. Okay, folks, time is up. If you have no more questions for me, you all have a great Thanksgiving. Don't eat too much and see you in December.